Tensor is not something casual. That is the product of the collaboration between two companies, Hamilton and Singleton. The two companies are in the top of their own technologies. Hamilton is leading the liquid handling automatizations, and Singleton is on the state of art of the single cells platforms. And we create together the Tensor. What is, let me show you what is our company doing. We are taking care from the sample, since the sample is extracted from the biopsy until the bioinformatic analysis in the single cells process. We have before automatized our single cells extraction. What is the secret of a good single cells experiment? To have a good single cell suspension. And we automatized our cell partition until the mRNA is captured. Now our Tensor is automatizing all the workflow from the cell suspension until library construction. We have also a tool that is very useful for bioinformatic analysis, our telescope. Our technology, maybe you know already or not. I will introduce you how is our base technology is the scope chip. That is 150,000 micro wells, where we are generating a laminar flow, and this laminar flow is making the cell partitions, and with the correct concentration, we are getting only one cell in some of the wells. And after that, we are covering the wells and the cells with our beads. The beads has a PCR handler for the later amplification. The barcode is unique for each bead. That will help us to identify every single cell in the later bioinformatic analysis, and the UNI that will help in the quantification of the gene expressions. The validity is capturing the mRNA. That's the basic information that you need. And you can see here how works our technology with microscopy. And here you can see one bead and one cells. The cells are much smaller than the beads, and only one bead can go in one cell. That is helping to avoid any not correct identification. After you know our technology, Tensor is a big instrument. Such instrument is very heavy, 170 kilos. We have a range temperature for working with the single cells and not affecting the transcriptomic. And the routine should be between 4 and 5 hours, depending on the number of samples that you are loading. Our sample rate is from one sample, if you need to run only one sample, because it's a very important sample that cannot wait for others. You can run it until 16 samples in the single cells process, or until 32 in the library construction. But you see, it's a lot of power what you need for running the instrument. That is because you have a complete laboratory inside the Tensor. You have a PCR amplificator, you have a thermoshaker, and we have several coolers for keeping the DNA or the cells in the correct temperature, or also our reagents. For sure, we have also UV decontamination, the cooling modules that I already mentioned, and magnetic modules that are necessary for the beads handling. Such a device needs three different controllers. The Hamilton run control is running the experiments, the UV control is running the decontamination control, and the maintenance. The maintenance is a very important tool that Hamilton has to ensure that the experiment each day will be successful, and also for keeping the warranty. After that, we incorporated 3 different processes and independent. That's very important. All the processes are independent one from other. The chip operation process, you are loading single cells, and you will get cDNA, purified cDNA. That is also a key point. You will run the quality control of the cDNA, and then you can make library construction. until 32, from 1 until 32 samples. And what is doing, what is the big benefits that you can get with our library construction? You don't have to normalize the samples. You set the concentration, the volume that you have from the samples, and the tensor will normalize for you. That is giving a lot of robustness to your library. In case your CDNA will not grant the quality control, you can run additional purification, or even the library can be resite selected. Also, maybe I can mention that can be done also with manual process samples. The Tensor workflow, starting with loading the machine, all the reagents and consumables that you need, and that takes for the chip process, that is the same single cells process, around 40 minutes, maximum 45, but it's in CAN, and will run 5 hours. For the library construction, it's even faster. The loading is around 30 minutes, and the library construction 4 hours. Then you are loading consumables and you are getting results. That is important. And these results, for sure, you have to run the quality control, standardized quality control. We can guarantee that there is no cross-contamination when you're loading cells due to our UV decontamination that we are making between experiments, and also when you have cells inside, you can be worried that the device can contaminate, but that is not happening. This experiment demonstrates that when we are loading human cells and mouse cells in all the different positions that we can have, that to ensure that the human cells is around mouse cells, you see that the gene expression for human is only in the human samples, and the mouse samples has only mouse genes identified in the bioinformatic analysis. I can show you also one experiment about the capture rate, what is one of the more important things. This experiment will show you 30,000 cells loading, 40,000 cell loading, 50,000 and 60,000 cell loading with 4 replicates from each loading. They are cell lines, and we make 32 library construction in the same run, and the input of the library were 50 nanograms, and the sequencing rate was 30 gigabytes. That is the experiment results. You can see the experiment one from the first day is 16 samples, 4 replicates from 32, 4 replicates from 40, 4 replicates and 4 replicates. And also in the second day, the capture range is pretty close in all the samples, and there is the resume. Let me comment the resume. The mean capture rate from both experiments is 36%, 40%. That is in both experiments. But if we look in details of each cell loading, our capture rate looks like decreasing when you are increasing the cell loading, but that is not happening. What's happening is that the probability of having 2 cells in the same is higher, meanwhile you are increasing the cell loading, and that is why it's some reduced. And the CV is very good. It's around 10%, what is giving you a good robustness of the experiment. That is different cell loading that we made. We handle from 2,500 cell loading until 60,000 cell loading. The capture rate is bigger with the smaller capturing, what is also good. Maybe we can go ahead with this kind of experiment in the future, and even loading smaller quantities that is expected to capture even more cells. And the variability of the capture rate is pretty good, always around or under 10%. The library construction also is not a contamination risk. When we are putting water on the cDNA, our library has 0 concentration. That means that the process is pretty clean. It's completely clean, and is not contaminating from one well to another. And that is the library construction. The library construction show with the tape station, and that's the profile that will have your library construction. That is the lower, and that's the upper, and that's the libraries. As you see, there is some variation, but the variability is more explained by the dilution of the libraries that explained by tensor related. The CD in 52 libraries is around. The main peak is variation is under 4%, and also for the fragment length is under 4%. We are very happy with the results. That is another experiment that I wanted to show you, that is with PDNC. We have 16 replicates running completely in Tensor, and that is another 4 replicates made manually the complete process by our expertise service teams. And I have to say that they are the MBA of the single cells teams. And that is an hybrid experiment. The hybrid experiment, we took part of the manual cDNA and we make library construction. That is overloaded on the replicates. And that is the separate samples. As you can see, always is the same cell suspension loaded in different chips. That is manual loading. That is manual loading in the chip, but Tensor library construction, and that is all the experiment running in Tensor. As you can see, the clusters are always the same, and the cell distribution is always the same, and the very, very small variation is a biological explanation, is because you are not loading the same cells in each chip. You are loading aliquots of the same cell suspension. We can see here, for example, comparing the cell type distribution. Maybe it's not too intuitive. I make some image. I was playing with the image. That is the manual, and that is the hybrid. But it's the same cDNA. I can show you. The same cDNA, loading manual, I'm making library construction, and that is the cDNA manual and library construction in Tensor. There is no variation between the samples. Also in the sample number 2, also in the sample number 3, and also in the sample number 4. And the live and all the that is I am showing you only here 8 of the library made with Tensor, and the complete 8 of the samples made with completely with Tensor, and the cell representation is very good. Here you can see is the same experiment, only the Tensor, the 16 sample running in Tensor, that 16 replicates, should I say. They are not different samples. It's the same sample with 16 replicates, and this is the informatic variance that we found is very, very accurate, should I say. And that is the genes that we use it for separating the clusters, and only with 3 genes, the main 3 genes that for creating the cluster, and you can see that are all of them perfect for the analysis. I would like you to make a short resume about the benefits of scaling to Tensor that will give you a statistical robustness, as I demonstrate with the data, increasing the analytic, and you can increase the analytical depth because your projects can have more replicates without more effort. Even you can make more different samples at the same time, but it is giving facilities for the experiment designs. Staying competitive, your workflow in the laboratory will be higher, it will be increased, and you don't need 2 devices for running the experiment. With only one, you can make both cDNA and library. You will save time. The complete workflow is around, let me calculate, around 12 hours, 11 hours. And on this time, you can make manually only 2 samples. And with this, you can make 16 samples. You will reduce the batch effect that is depending on which technician is running the experiment. Then I think it's a good product. And let me know if you have additional question. I will try to answer, or I will try to find anyone that can find the answer for you. That's all from my side.
Thank you, Juan, for the presentation. So one that arrived early on in the presentation is which method can separate just one cell into the bead? Yes, we can go back to the slide maybe. Yeah.
Yeah, here you can see this barcode. There is several millions of these DNA monocatena DNA construction, but this barcode is unique for each bit. And as you can see, one bit is on one cell, and then all the mRNA that is captured from this cell will have the same barcode. Thus, we can, in the bioinformatic analysis, we can read like that. We can interpret that all the mRNA with this barcode is what is coming from one cell. And also we can quantify how many copies from each gene is expressed, because the UMI is giving us is also is unique for each line. That then each mRNA will have his unique UMI per cell. You can quantify how many copies, even if the PCR is amplifying the same cDNA.
So the next question is, would it be possible to make library construction from single cell and single nuclei cDNA simultaneously?
Yeah, good question. Thank you. Single cells and single library and single nuclei. You cannot run the experiment, the cell partition, and you cannot run this experiment simultaneously in the machine. You have to run separately. But once you have the cDNA, you can make the library construction at the same time. You can make that because the tensor will normalize the cDNA for you. You don't have to take care about that. You have to just note the concentration in the Excel table that you have to provide when the tensor is starting. You have to provide the cDNA of each sample, and each single sample will be diluted will be normalized to 10 nanograms or 50 nanograms according to your choose.
Yeah. And another question, could you please provide the publication behind this equipment? So behind Tensor, I mean, Tensor is a very, very new launch, so.
A publication, currently we don't have any publication. We have laboratory data. We can provide you laboratory data from different kind of samples. We are running also tissues. We are running also experiments from some customers that want many replicates. But this kind of information is, we cannot provide you information from our customers, but they will publish in some year, I think in 1 year or something like that. As you know, a paper takes time, and the launch was, I think, 3 or 4 weeks ago, and now we are making a webinar. Then the first publication will be expected, if it's a very good experiment, minimal at the end of the year.
Yeah. But meanwhile, we can share some data from our internal experiments, and we can share other publications on the scope technology. We can contact you right after this webinar with some more information. We can send an email and continue the conversation. So for whatever question you have, we can also follow up after this webinar. So that's great. And I have one last question. Meanwhile, I would encourage everyone to send some more questions because we have plenty of time. But for now, I have one last question, one extra question. What's the number of samples that can be run with Tensor, which is something that we touched a bit earlier on, but we can always mention.
Number of samples. You can run from one sample with the single cells process. You can run from one sample until 16. If you have 3, you can run 3. If you have 14, you can run 14. If you have 11, run 11. And something important also for this kind of experiments, the reagents that you need will be calculated for the number of samples that you are running. You are not discarding all the reagents or all the chips that you are not using. If you are using 4 chips, you will put 4 chips and not 8. And also the reagents that you need will be the required for this number of samples. Then from 1 to 16 in single cells experiments for single nucleus, and from 1 to 32 for library construction.
Yeah, thanks, Juan. Another question, did the cell isolation kits from different brands affect the efficiency of the equipment? That's a good question.
If your cell suspension is a good cell suspension, will be not affected. For sure, we are not using external kits, but if you have a good cell suspension, for example, we are using PDNC insulated for external customers, and it's working. Then, if the cell suspension is a good cell suspension, will be not affected. What is affecting is the viability of your cell suspension should be over 75, 70, 75%. Under this, you are sequencing information that is not bringing you nothing. It's not because cannot be run, it's because you will have not good information and you will pay the sequencing of this information that is not bringing nothing for your experiment. That's why we insist always in using over 70, 75% of the viability.
So then thank you all for being part of this webinar. Thank you, Juan.
Thank you. Ciao.